Okay, so first of all, thank you for all the speakers for the great and exciting presentation. We are going to move now to the panels, and we have a great lineup of panelists that will be joining our speakers. Yeah, so basically we have Patrick Wen from Farber that will be joining, Lauren Abrey from Novartis, Uri Weinberg from Novocure, and Kirk from the National Brain Tumor Society. And starting with the panel, so Lauren, I would like to start with you. We heard from Steve Bagley about the opportunity of using liquid biopsy to improve clinical trials and drug development. I'd be happy to hear your thoughts about the pharma perspective about the barriers in drug development for brain cancer and how liquid biopsy can maybe improve the efficiency of some of these programs. So the question is to you.
So thank you, and thanks to all the speakers. Like everybody else, just a quick check. You can see and hear me okay?
Yeah.
Terrific. I think really elegant science, really nice progress in a difficult space, and I think the speakers have done a lovely job of highlighting, where the opportunities and challenges sit. I think if I flip myself fully into kind of, where I sit today in a large pharmaceutical company looking at drug development, and, people wonder why or why don't we look at brain tumors, I want to go back to kind of something really simple, right? My wish, if this could take off, is that we would use circulating tumor DNA, whatever analyte you decide to use, to really develop drugs intentionally for brain tumors. And, I put a little note in the chat to Howard Scher when we joined the call. And I remember that when I left Sloan Kettering, I did a piggyback study on one of his prostate cancer studies because it was the only way we could get access to drugs to try to target the PI3 kinase pathway in brain tumors. But in fact, we had no idea if we were doing anything. So I think, aspirationally, this would not only be a way to identify patients, but potentially to do pharmacodynamic monitoring so that we could really develop drugs that are intended to treat patients with glioblastoma, not kind of use drugs that were developed for another cancer and try to get them to work, which I think fundamentally is a flaw for really developing transformative therapies in this space. So I see that as kind of my aspirational goal. I think in the kind of practical short term, I think it will be really important to standardize some of these efforts, because today we still sit with drug development and a lot of concern about interpretation of the imaging. So if we could have something that gave us more confidence in the imaging results so that we felt confident that a response was a response and we didn't argue about pseudo response, confident that progression was progression and not arguing about pseudo progression, and only relying then on overall survival, that would be a huge win. Again, I think it's going to be a big ask, right? I think there's still a lot of need for improvement there. So those would be my biggest wishes for what we could achieve, but also where some of the barriers sit today when you think about how companies, particularly large companies, are likely to be skeptical of this space, where there have been numerous trials that have really largely been negative. I think there's a great opportunity for small biotech to make some huge progress here if they partner smartly with some of the scientists, as we've heard today. So that's my first thought, and very curious to hear what the other panelists think in the same area.
Yeah. So I can take it from here maybe. So hello everybody, and thank you to BLOODPAC for the opportunity, and of course to the speakers for their fascinating talks. So I think I'd like to tackle this from a very practical perspective maybe. And I'm serving as Chief Science Officer for Novocure, and we're dedicated to the development of tumor treating fields, or TT fields, which are basically alternating electric fields at an intermediate range of frequency that act as an antimitotic therapy. And tumor treating fields have been approved for the treatment of newly diagnosed glioblastoma following a phase 3 study that demonstrated extension in overall survival, and we already have an NCCN category 1 for that. But throughout the development of the modality, there are many critical elements that we could integrate into the development of this particular modality in cancer, and some of these are unique in the field, and some are similar to maybe other local or systemic therapies. For example, we have the ability to potentially adjust or fine-tune the electrical properties of TT fields in order to make them even more individualized for the specific tumor and specific patient. And using liquid biopsies to get fast input regarding that prior to having imaging studies and maybe less direct feedback is of great importance. We could basically attempt to do this fine-tuning while the patient is on therapy and increase the likelihood of successful therapy. In addition to that, we are treating the entire patient population in newly suffering from newly diagnosed glioblastoma, and nevertheless, we are, despite being a commercial company that would like to treat a large patient population, we would like to to also identify those patients who could benefit from it the most. And this method, as discussed, could facilitate that and maybe identify characteristics or even helping defining a biomarker that would facilitate the pre-treatment identification of the relevant patient population. One other thought, and I'll finish with that maybe, is that we know the more we treat, the better the outcome from the therapy. But perhaps for certain cases and patients, we don't need to encourage them to have to maximize the treatment duration. Maybe we can define the threshold for a minimal duration or minimal usage of our modality that will still secure a response from the treatment. Of course, diagnosing decreasing sensitivity, again, facilitating fine-tuning of the treatment parameters. All of these are fantastic opportunities for our modalities and for other modalities that will really improve the development of the therapy for patients.
Okay, thank you, Laurent and Uri. If I'll be happy to also forward it to the other panelists and speakers if anyone want to address. So basically, I assume there are larger and larger pipeline of candidate targets, including immunotherapies and targeted therapies that are going into the clinic and into clinical trials. And if you have any thoughts about how to design clinical trials with liquid biopsy in order to do quicker, more effective selection of which candidate are actually working before doing that on hundreds of patients over multiple years, which basically how to optimize dosage in a way that will be effective for adults and especially for kids. So any last thoughts before we move into the next subject, if anyone wants to address?
Csf, did you want to talk about some of the challenges, or when you're saying the next topic, are you talking? I just wanted to, where do we stand here? Want to think about regulatory issues or the big pictures there? So
yeah, basically we are happy to jump forward to that question if you want to address it. So some of the challenges are not necessarily technical challenges, but there are a lot of challenges that we heard before about the code that is required, the consortium of multiple cancer centers, regulatory issues, and so on. So yeah, if you want to address some of these, we'll be happy to hear.
Sure, no, but great. And so, I mean, first of all, just great presentations all around and great discussions. And, many thanks to Lauren and Susie and Csf for giving me a chance to be a part of this conversation. I think it's really important. I think it's really timely. And I think, these challenges that we're talking about is, we should really welcome them because, I think, in talking about are there drugs or, how can we use liquid biopsy? We have to be confident that we actually have clear hypotheses and, both in terms of therapies and how diagnostics can work in brain cancers. And so I think, I think the good news is, and I'm more and more confident of this all the time, is that we're almost there, or we're there, or we're close. And so the key now is to really figure out, how we can be efficient and rational in how we implement this. And liquid biopsy for diagnosis across the spectrum would be really important from that. I know from my vantage point at the National Brain Tumor Society, what we have is we have a really motivated patient and caregiver community. And they're open and committed to clinical research. So then when you combine that with neuro-oncologists, neurosurgeons, translational scientists, you've heard from today already, who are willing to cooperate with one another, you really have a really powerful group of people to move things forward. You have all the stakeholders. And so that's really important because then putting all that together, you have to then figure out how to leverage all that and work together in partnership. And, again, that's been a reoccurring theme is the partnership, the networks, and figure out how to work with the regulatory agencies, the FDA, the EMA, and to really efficiently validate and bring forward this new diagnostic thing in the brain tumor space, which has been so beneficial across other solid tumors. And also then think about how you're going to utilize that to guide therapeutic options for patients with brain tumors. One other thing is that there's a money component, right? There's a critical capital investment, and this goes to Lauren's point on the big pharmas maybe a little bit shy of this, but the good news is that big pharmas are also starting to get back a footprint in brain tumors. But where's the money component? Where does the funding come from? And so I think there as well, we need to be creative and really foster relationships and partnerships between traditional capital funders like venture capital, but also venture philanthropy, foundations, family offices, and government funding agencies. And all of those together is really going to be how we're going to move this forward. I think one benefit that we do have collectively is it's also sadly is that the bar is relatively low, for brain tumors like glioblastoma, DIPG, ATRT. Ultimately, at the end of the day, your cohort size for demonstration of clinical value may not need to be that large. However, you do need, again, the theme of being able to generate a lot of data and share the data on liquid biopsies, I think is really important. So, as I think you all know, additionally, there are other regulatory constructs to improve patient access, like the FDA breakthrough designation has been mentioned, but also in particular in the pediatric space, there's also potentially priority review vouchers. So I think, we can expand on all these points, but just one last thing I wanted to mention before closing was, I think we have a real opportunity now with the current administration. I think we need to really work closely and support President Biden's commitment to oncology and to brain diseases, and hopefully to, really focus in on brain tumors and really explore ways to leverage and accelerate this for the benefit of our patients. So I'll stop there.
Great. Thank you. Thank you, Kirk. Any last note, and again, addressing more to Patrick Quinn and the other speakers regarding the opportunity of incorporating molecular monitoring in the clinic, comparing that or complementing the current imaging-based surveillance. How do you see the benefits of using imaging with liquid biopsy together, CSF versus blood? If there are any last comments about that before we are moving forward. So Patrick,
Thanks so much for having me. And we're so grateful that you guys are looking at this topic for brain tumor patients. It's hard to imagine another cancer that needs liquid biopsies more, and yet we have more challenges than most other diseases. I think the other thing, in addition to issues like sensitivity and specificity, because our tumors are so rapidly growing, the time for doing the assay and getting the results back is perhaps even more crucial than many other cancers. So that's something for the scientists to also take into account. I mean, I think Steve did a really great job in highlighting many of the areas where there are needs. I mean, diagnosis, separating lymphoma from glioblastoma and metastases is such a common problem. And the MRI doesn't tell you the difference. And so if there was a way to do a liquid biopsy quickly, that would potentially mean the patient doesn't have to go through unnecessary surgery. There are common scenarios where patients present with a flare signal, and you can't tell if it's MS, multiple sclerosis, or a low-grade glioma. And if there was a liquid biopsy looking, for instance, at IDH mutations, you could make the diagnosis, and that could save patients from unnecessary surgery. But I think the major area right now that's been problematic is the issue of pseudoprogression that a lot of people have mentioned. This occurs after radiation therapy in 30 to 40 to 50% of patients. And actually tomorrow, the National Brain Tumor Society is hosting a RANO session where we're going to talk about the updated RANO criteria with the FDA. And basically imaging in the first 3 months after radiation is completely useless. You can't tell anything during that period. And so if there was a way with the liquid biopsy to show that it's tumor progression as opposed to radiation effect, that would be huge. And then as we develop immunotherapies, the issue of pseudoprogression is likely also to be problematic and could be helped with liquid biopsies. I think one of the things that as a field we've shied away from is taking spinal fluid because it seems difficult, it's hard to put the patient through it, and sometimes it's dangerous. But actually changing that mindset, if it really is effective in helping us get an answer, it's actually a much easier procedure than a bone marrow biopsy. It's not as painful. And as a field, we could change the mindset and perhaps do CSF examination more commonly. And then the final area of huge need is the temporal heterogeneity in these tumors. So for instance, one of the major targets in glioblastomas are EGFR mutations. And they are lost in about 50% of cases at recurrence. So this year, there are 2 and possibly 3 trials of new EGFR inhibitors for recurrent glioblastoma. And right now, those patients are going to be enrolled on trial based on their original genotyping of the original histology at diagnosis. And half the time, those mutations may not even be there at recurrence. And we're usually not going to rebiopsy these patients. So having the liquid biopsy in that situation would be really helpful, I think, for all these targeted therapies and potentially for other therapies as well.
Thank you.
Patrick, if I want to jump in, and I just would echo what you're saying, because I think the risk there is huge that we potentially then discard a therapy that could be effective if it really did have the right patient selected. But if you're selecting from diagnosis, treating at first or second recurrence with unstable genetic mutations or genetic alterations, we don't know what we're seeing, right? So, and I think your data and our data in some fashion, looking at the small subset of patients with V600E mutations and showing that we can have an effect with drugs like, whether it's TAF-MEK or other BRAF-MEK inhibitors, is a good proof of concept that we can get past this. And that is a stable mutation that typically won't change over the course of the tumor's life cycle, let's say. But I think those are really critical. And I think the other one you kind of argued about or highlighted, but I think is critical for this field, is that transformation of low-grade glioma into high-grade. You know, we follow these serial scans. We're never quite sure when it's transitioning, but if you could get ahead of that to optimize kind of when you want to introduce therapy, I think for patients, that would be enormous for the benefit. So.
Yes, thank you, Lauren and Patrick, for those really important comments. And I'd like to ask, we have a question in the chat, what's the risk of a lumbar puncture versus a bone marrow biopsy? And perhaps Nir or Manmeet, you could put it in perspective. What's the risk of a lumbar puncture in general for the audience? And also, in either of your studies, are you comparing the post-focused ultrasound blood-brain barrier opening cfDNA levels to CSF as well? Or Nir or Manmeet on?
Sure, I can address that, and I'm sure Nir will have more things to add. So, a lumbar puncture in someone who doesn't have a large mass effect is a fairly safe procedure. We do it all the time in patients with brain metastases, where we are trying to see what's the leptomeningeal spread, and that at times impacts therapies there. We don't tend to do it very regularly in primary brain tumors because, we kind of continue that in the same space, and our therapies, as was alluded to, are fairly limited to begin with. But can we do it? Absolutely. Do I agree with Patrick about, it being a relatively safe procedure as a BMT, and as one of the ones who has actually done both, since I'm a medical oncologist, and I can just say it depends on the type of the patient. But if you have the expertise, both the procedures are fairly safe and with very minimal side effects. I think people in heme malignancies have done a phenomenal job with bone marrow aspirates and that guiding therapy all the time. And they have the drugs, right? So I think as we're getting more and more drugs, I think this is going to be critical, how we use this going forward. And Patrick alluded to, a couple of trials that we have been involved in as well, looking particularly at the EGFR space in terms of therapies. And I think it also depends, what's the therapy that is being used. So if you use EGFR-directed therapy, then yes, there's almost a 50% chance in change of the EGFR because of escape mechanisms. So I think it's going to be critically important. Now, as far as your second question is concerned about the trial, the ones that we are doing in that, we are just looking at plasma sampling, and we are comparing more for, comparing that to the tissue and at different time points. But definitely CSF, presents an exciting opportunity, but I'll leave it to Nir to see if his or his group are doing that.
Yeah. No, thank you, and thank you, Susie, for the question. I'm just echoing what was said. LPs are generally quite safe, and I think that it's important that we put things in perspective. We're talking about a spectrum of invasiveness here, and when we're talking about neurosurgery, which is the ultimate sort of risk, that patients take and that surgeons take when treating patients. That's sort of the standard, and we need to compare, to that as well. LPs are certainly safe, but they're cumbersome to perform, and it's hard to convince or to justify multiple LPs serially over time, divided by several weeks and months, et cetera, with patients. Obviously, it's done in trials of neurodegenerative disease and neuro-oncologic applications. But I think, if we get a signal, and if we start detecting a signal, and this is where, the collaborative work that, BLOODPAC and other sort of collaborations can really come to its own, then we may not, have to go that way. And I think that, to Manmeet's point about the trial that's happening, we are looking at plasma before and after. And we are comparing to tissue obtained at surgery. And importantly, focused ultrasound, for example, being less invasive, image guided, we have that imaging time point there to directly compare and to provide confirmation about pseudoprogression or actual progression. So I think that it's important to double down on investments on the kind of samples and enriching the kind of samples that we get with plasma. And seeing sort of what the yield of that would be over time.
Okay, thank you for everyone. I think we had a very important discussion. Again, we wanted to hear also all of the perspective, also from the pharma opportunities and challenges, and basically arranging clinical trials in a larger scale. I would like to address the last question actually to Lauren Lehman and to the general BLOODPAC audience. And basically, BLOODPAC have been taking a leading role in the standardization, data share, establishment of clinical industry collaboration, liquid biopsy. We are hearing that basically there is both a lot of challenges in free analytical standards, sample collection, generating basically the clinical consortium that are required, and also data share between organizations. How do you feel that BLOODPAC can help to promote the incorporation of liquid biopsy in brain cancer? What is the opportunity and the impact that we can make?
Sure. First, I'm going to invite everyone to turn your cameras back on so we can all see one another. And try to encourage a fun dialogue here. There you go. Look, Maddie's back, making me feel better already. Okay, good. All right. You know, again, I'm so excited to hear everyone and the enthusiasm from everyone and so many questions, which has been wonderful. You know, I have seen and I have suggested from day one 3 kind of easy ways forward that we have done in the past. You know, I think there are a lot from my discussions with these wonderful speakers and panelists. There still seem to be BLOODPAC sweet spot, I think, still remains in the setting and establishment of frameworks and standards for the liquid biopsy community. And based on my conversations, it still sounds like, and I think Stephen actually wrote in his presentation, standards for biofluid collection for brain cancer. So, you know, I think we'd be a great partner to actually bring together all the stakeholders in the field to help work on setting some of those standards. I think that's an easy working group under our recommended data element program. And Jake's here, co-chair of that group, to actually work on. Bob Grossman, I think, is also on the call. You know, from my second, I think an easy box to check is we've also collected, and Don's here with us too, worked on a project called Project Exhale in the past, and we're still continue to work on it, which is looking at liquid tissue concordance in lung cancer, retrospective studies that have been collected from our members that have made donations of that data. I don't see, you know, I think doing a project like that would actually be interesting if we were able to aggregate data into the data commons. Would be a very cool idea. We can ask Bob his thoughts on that too. I think everyone put in their studies, the idea, their presentations, the idea of a prospective study, which I think would probably fall into the category of one of our pilot studies, which we have run in the past. We have essentially 3. One that was funded by, it's kind of seed funded by the Prostate Cancer Foundation, one that was seed funded by the Breast Cancer Research Foundation, and Don Johan study that's funded by the FDA. So looking at prostate, breast, and lung cancer. And the way we've been able to run those pilot studies have been our academic members from those studies receive the seed funding from those organizations, and we're able to identify interest among our corporate partners to provide in-kind support. So you're kind of able to leverage the partnership within BLOODPAC, and anyone who participates in those studies is a BLOODPAC member. So I think, and we have explored the idea, I should say, in the past of a pediatric pilot, it's been discussed. We know that there's interest in that, and I think there is obviously interest in brain cancer for adults as well, although we haven't had a specific discussion around that. So I was thinking that maybe those were kind of my very upfront, easy to mention ideas. I'm just going to bring on my big guns and maybe ask, for starters, Jim Godsey, who I think is on the phone, maybe Kelly Bramlett, and I'm not sure if Bob is on with us, and Don, to kind of give us some ideas. But Jim, I know you're on the phone still, and maybe you have some thoughts.
So first and foremost, just congratulations to all of the researchers, the speakers, presenters. Amazing work, missionary work. And you think about the single-digit overall survival of glioma patients, and being able to from the perspective of get therapy in past the brain-blood barrier, and then be able to use ctDNA testing to vastly extend the diagnostic capability, and ultimately marry the two, precision medicine. So a couple of comments. So I loved the work that C2I was driving with low-pass whole genome sequencing. We've seen how breadth versus depth really does enable very sensitive, specific testing. And I wasn't aware of the progress there. So I love that aspect of the talk. But then, from the focused ultrasound perspective, the perspective that you can, in fact, reach every aspect of the brain, and the roughly threefold increase in cell-free DNA, concentration versus post versus pre. It would be great to translate that into, say, diagnostic yield. So is there a significant increase in diagnostic yield with FUS treatment versus not? And then the fact that they're able to get large molecules into the brain with the high intensity treatment versus now perhaps match that with molecular guided precision medicine with cell-free DNA. To see that type of study design would be, I think, fantastic and very important, for future FDA engagement.
Thanks, Jim. Any questions for Jim? I know it's kind of awkward because he's not on camera. Love the camera. Kelly, I'm staring, you're right front and center in front of me. I'm not sure if you have anything to add or thoughts.
I would just kind of echo the same things. Really good discussion and good topics. I do think so I was the one asking questions in the chat. I think my name is kind of awkward, so you can't tell it's exactly me, about RNA evaluation or gene expression evaluation. I was kind of thinking about it in terms of the whole genome sequencing and kind of the information you're getting there from the liquid biopsies, and if we've looked at RNA expression there. So I definitely think there's stuff to do. I think BLOODPAC is a good place to maybe define start defining some experiments that would be useful for many people in the field and moving the field forward. I am interested, and I haven't heard yet, if we got a response to Jen Dickey's question about this focused ultrasound, and has there been any evidence that it would kind of, negative side effects of this? We keep saying it's a safe way to open the BBB, the blood-brain barrier, but what about, does it help with metastasis or anything like that? Or does it have any negative side effects that we know of yet that you guys, or what do you think about, worry about, I guess?
Thanks for that question. It's so far the safety data is fairly compelling, but it's under active investigation. Obviously, the more novel indications that we treat, the better filled out and more comprehensive the safety profile becomes. All patients that get focused ultrasound for our secondary or metastatic tumor trials are all on active chemotherapy, which would reduce the risk, we think, for metastases. These patients have very well controlled systemic disease, and we follow them for months and in some cases years after, and we have not found this to be linked with the development of any new systemic disease or actually new even intracranial disease in the patients that we have treated so far. You know, when you look at side effects and adverse events, you can obviously look at things clinically and radiographically. Clinically, these are very well tolerated procedures. So patients go home the same day. They're followed very closely, but we have not found any serious adverse events. The adverse events that we found mostly are related to things like discomfort with the frame or prolonged time in the MR, et cetera, which are all improving with time. Radiographically, we have not found this to be linked with any significant, again, serious adverse events, whether it's bleeding, swelling, hemorrhaging, or anything like that. So it looks like it's quite a safe, well-tolerated procedure so far, but additional data is obviously being collected prospectively.
Yeah, thanks for reminding me that you showed some really fantastic photos of the actual, I don't know, let's call it a helmet, stereotactic implementation that is used, which was interesting to help us all wrap our brain around what that looks like. That was, I enjoyed that. So thanks for sharing that.
Yeah, the fact that it's image guided, you know, and actually is on the table is one of the value adds here. I mean, I think, you know, what we envision potentially is, you know, as Stephen showed in his talk, you know, a patient comes in being investigated for some kind of neurologic symptom or headache, and they get a scan and there's a lesion. The fact that, you know, the helmet is coupled to the MR allows us to develop a kind of point of care non-invasive biopsy, essentially, of this. And the better tools we have to assay peripherally, the more valid that becomes. So it's the pairing of ultrasound with imaging that really is where the power of the technology can come in.
Yeah, very, very interesting and cool. Thanks.
Thank you.
I don't like silence. What other questions do we have? Howard, you coming in?
I don't know if there's it's a question, but you know, I want to thank Lauren for a little memory here. You know, again, you seem to be exactly where, or not exactly, but where we were in terms of the pseudo progression question relates to bone scan. Obviously, yours is much more difficult than we were looking at because we were just looking for more spots. But you know, I think the work is really interesting. It's excellent. But I would only suggest, and I know you're sensitive to this, is don't try to bite off too much with each question. You're in the early clinical validation stage, and the more you can do to standardize interpretation of the MRIs and really try to identify those features which, you know, try to better separate out it. It's going to be, you know, again, a challenge, but I think it's doable. And you really have an opportunity to transform management here. So I'll say welcome to the party and look forward to working with you. I should also let you know that our group, our neurosurgery group and radiation oncology group is also very excited to join you, but had conflicts this afternoon.
Right, Susie, back to you.
No, I want to thank everybody for coming on. I see we have Phil Thiebaud on and Bob Grossman, and I know you always have some really good opinions and suggestions, so maybe you could chime in a little bit.
Well, I'm certainly happy. I thought the presentations were outstanding and fabulous science. I do remember when that Batagawa paper came out, how disappointed I was to see gliomas towards the end and having so little cell-free DNA. It's quite, it's exciting to see how much more we understand now and how we see opportunities where there is a signal, or through focused ultrasound, you can amplify the signal. I think with respect to what we're doing at BLOODPAC, really the MRD space is going to be so critical because, you know, the data we saw today is very compelling, very aligned with what has been seen across different tumor types. And certainly for us to make meaningful progress in the therapies for these children and adults with brain tumors, we have to get much more facile with how we sample the tissue. It's been a major limitation because of location. And what I saw today got me very excited that, you know, the investigators and the individuals here are really driving that. And through that, you know, appropriate combination of sampling the blood, perhaps intervening to break down the blood-brain barrier in order to amplify the signal in the blood, there's going to be a real opportunity to find a path for new therapeutics, assess them more rapidly in that era, in that time period that Dr. Wen suggested, where imaging cannot be helpful because of all the changes that are a result of the therapy. We've seen that again and again, immunotherapy. Howard mentioned prostate cancer. You know, if you can get that cell-free signal, it can really clarify that early response, early resistance in a way that few other technologies can. And so it's very exciting today. Really good data suggests showing that you have to go broad to understand the signature, that the panel approach isn't working as well. We're seeing that as a consistent theme. But great work, exciting, fabulous to have you guys considering participating. I think a lot of what's going on in the MRD group could work really well with this.
Don, I'd love to hear from you.
Sure. I would just say that the importance of the BLOODPAC data commons and being able to harmonize the data, liquid biopsies compared to, you know, still the current gold standard, which is solid tissue biopsy and tumor analysis, is really important. And, you know, working with, you know, having the good fortune to work with Bob Grossman, who's designed all of the NCI data commons and, you know, setting things up and also correlating things with eventually with clinical data, as well as all of the different modalities that we can do now with high throughput next generation sequencing is extremely critical and should help us arrive at these answers quicker, especially as we look at more and more novel techniques like we've had at this session today. Thanks.
Well, thank you. And I appreciate all your comments. And I see Dr. Akral in the audience, who is also a pioneer in this field of liquid biopsy and brain tumors. Maybe you can give us your opinions and suggestions.
Oh, sure, Susie, thank you. I appreciated being a part of this audience and listening in here. I agree completely with Steve Bagley and Patrick when how they framed it in terms of the need and what we've run into in terms of the sensitivity limits of doing cell-free DNA analyses in the blood. And I think Manmeet and Steve both pointed out that the clearance of cell-free DNA has become really quite interesting, both for MRD, but also for calling response to immunotherapy and pseudoprogression, which is, as Patrick pointed out, a real issue, is that we see these tumors grow, and how do we not call them PFS, and how do we sit on them, and not having a good readout in the paper that Manmeet shared, where we can see across 25 different types of cancers that we can accurately predict response to immunotherapy using liquid biopsy is promising, but our issue is yield for the brain tumors. And so I think the work that Nir presented and Manmeet covered also the work that Nir and Graham had done, showing that we could boost that signal up to 7-fold in Nir's paper is really quite meaningful. And we're hopeful that this trial, and Incytec is funding a pivotal trial to try to prove out what Nir and Graham saw in the safety and feasibility setting at a larger number of patients to see if we can have an indication for using focused ultrasound at low intensity for liquid biopsy enabled by focused ultrasound. And so all of the institutions represented here are leaders that we're engaging as part of that trial. And we're hopeful, as Manmeet mentioned, that process that Nir highlighted could also be synergistic, not just to get more therapeutic to the tumor microenvironment, but we're seeing that this liquid biopsy signal that is released from the region also includes a neoantigen release, and there's an immune priming response that happens with the BBB treatment that's synergistic with the therapeutic delivery itself. And so we're getting sort of a read-write process where we're getting more agent to the microenvironment, more neoadjuvant release and immune priming, turning the cold tumor hot, and we're getting signal, region-specific signal back in the blood to track that treatment. And focused ultrasound can just be layered in as a monthly treatment. And one question was about the safety, and Nir talked about that. We had to summarize just our experience with the FDA recently, and it's now been over 150 patients treated with focused ultrasound using the Insightec device in over 400 cycles of treatment, led by Nir and the others on this call across a number of trials from Alzheimer's all the way to brain tumor space, and zero serious adverse events and no treatment-related events related to the BBB disruption. So it's really a nice top-line safety signal. And the key really now is to focus on clinical benefit. So we're excited to support these trials at all of these institutions.
Great, thanks for that comment. We have one last question in the chat that I would like someone to address. It says one of the advantages that ctDNA from blood has from a developer standpoint was the easier availability of samples from biobanks and vendors. Is there a similar one for CSF? Anyone answer that question?
I can comment from our experience. We have a program for banking the CSF, but it's a very precious resource, so we don't like giving it up, and I don't think anybody else would be easily giving it up. There's way more blood that gets banked, particularly in adults, but I would say CSF, particularly for pediatric brain tumors, it's a very scant commodity for anybody to give up.
I was going to say one of the advantages of having BLOODPAC together is the diversity of experiments and research and development that was done when BLOODPAC first started. There were groups that came together and everyone was able to share data and say, yes, we believe that this blood collection tube with stabilizing agent is equivalent to another method that was widely adopted, and therefore we don't have to continue to do stability studies on these. But they weren't doing those with clinical trial samples because of the risk. They were using commercially acquired material. And I think that's going to be a barrier for building standardization within this field for CSF. So clearly there are vendors that create extraction products and their protocols are written, but using the collective experience of something like BLOODPAC is going to be more challenging because I bet there aren't too many experts within this group in brain cancers and CSF.
Okay.
Yes. Hi, I'm Alex Miller. I work at MSK, and I've been doing work on CSF liquid biopsies, primarily for glioblastoma patients. So just to speak to the biobank or the CSF bank, we also have a collection of CSF of about 300 samples in the adult space and about 200 in the pediatric space. And of course, it's obviously a valuable resource, but we found that, even though glioma patients don't typically get tapped for diagnostic purposes, when they're admitted to the hospital and on service, they are often tapped. So just by banking those additional samples, it could be a way to build up a bank, I think, across institutions. And you actually don't need a very high volume of CSF. Most of our work has been done on 3 cc's of CSF, and you typically take about 20 off of an adult patient when you do a tap. So I do think it would be possible to build one up if we did a focus on that.
Okay, thanks, Alex. Thanks, Jonathan. We've gotten to the end of our time. I want to thank everyone for listening to all of these expert speakers and panelists and all of your participation for today. We brought together industry, academia, and nonprofits in hopes of working together to expedite research and development. I want to thank Lauren for leading BLOODPAC and supporting this event to address the needs of this underserved population, and Asaf for his incredible vision in creating this agenda for today. I want to remind everyone that this topic is super exciting, but you have a whole day tomorrow that you can listen to more about liquid biopsy at the summit, and Lauren has put the registration link in the chat. So tell you and all your friends because everyone can join that. And Asaf, do you want to make any last minute comments?
So first of all, I want to say that I believe that we are on the verge of very exciting shift in cancer management. I think the data that we showed of basically using low blood collection volume and integrating coding and non-coding features across the genome allow us to really go into cancer types with low mutation load, pediatric cancers, things that were basically seen as a very low success rate before. I'm very excited about what we can accomplish in the next couple of years. Looking forward to working with all of you. And that's it. Thank you for everyone that joined the meeting today, to the speakers, to the panelists, and all of the audience. We are going to create a white paper that will be available in the next coming weeks, and we are going to post also the video recording of this event, so stay tuned. If there is any additional questions, please feel free to contact Lauren, Susie, or me. Our emails are going to be on the chat, and thank you. Have a great day.